So before I get started with RNA splicing, I want to point out that there are some modifications to the mRNA. So at the 5' end, there's a modified form of guanine which creates a 5' cap, and at the 3' end, an enzyme adds many adenine nucleotides which form a poly A tail. So with these modifications, it allows facilitates the export of mRNA from the nucleus. It also prevents mRNA from being degraded by enzymes, and it also facilitates the attachment of mRNA to ribosomes. So in RNA splicing, large portions of newly synthesized RNA are removed, and these removed portions are known as introns, and the remaining portions left after the removal of introns are known as exons, and they're spliced together by something called the spliceome. And now a spliceome is a large complex made of proteins and small nuclear ribonucleic proteins, also known as SNRNPs. And this plays an important role in the removal of introns and the joining together of exons. So, catabolic RNA is known as ribozymes, and SNRNPs contain snRNA, and this will attach to the intron-exon junction through base pairing, and the snRNPs acts as a ribozymes, and it removes the intron and it leaves it degraded, and then the exons are spliced together by other proteins, and this leaves a new mRNA. And the poly A tail allows from the movement of mRNA through nuclear pores. So this diagram represents enhancers and transcription activators. So within the DNA sequence, activators will bind to 3 sites which are known as user control elements, and they are found within the enhancer. And in the promoter within the DNA sequence, there's a Tata box which contains a sequence of thymine and adenine, and it is very crucial for forming transcription initiation complex. So a DNA bending protein will bring the activators closer to the promoter. General transcription factors, mediator proteins, and RNA polymerase II are nearby. So once the activators are bound to the enhancer, they then bind to mediator proteins, general transcription factors, and then ultimately they will bind to RNA polymerase II, and this allows for correct positioning over the promoter, and this creates an active transcription initiation complex, and this whole process increases the synthesis of RNA. This diagram represents regulated synthesis of repressible enzymes in the TRP operand of E. coli. So in E. coli, this TRP operand is usually on, and it is on because the repressor is inactive. The code for this repressor comes from TRPR, which is a regulatory gene. So since this repressor is inactive, it cannot bind to the operator due to its shape. But RNA polymerase can bind to this DNA, and it will continue the synthesis of RNA. And with the synthesis of RNA, it will create these polypeptide subunits, which will code enzymes for tryptophan. However, too much accumulation of tryptophan will cause tryptophan to act as a co-repressor. And when it acts as a co-repressor, it binds to this inactive repressor, and once it binds, the repressor becomes activated. Now that it is activated, it changes shape, and it changes shape because it is an allosteric protein, which means it has an inactive and active stage. So now that the shape is changed, it can now bind to this operator. So once it binds to the operator, it inhibits RNA polymerase from binding to the DNA, and it inhibits the production of RNA. So without the synthesis of RNA, these polypeptide subunits are not created, and tryptophan has just inhibited its own production. And this is negative feedback, and this is an example of how gene expression can respond to changes in the cell's internal and external environment. So this diagram shows the lac operon in E. coli for regulated synthesis of inducible enzymes, and it is an example of negative gene regulation. So an enzyme is labeled as inducible when its synthesis is induced by chemical signals. So LAC-I, which is a regulatory gene, codes for this repressor, and this occurs with the absence of lactose. And so the active repressor binds to the operator, and since it binds to the operator, it prevents RNA polymerase from binding to the DNA and going down the length of the DNA. And when this occurs, there's no synthesis of RNA. However, when lactose is present within the cell, allolactose, which is an isomer of lactose, acts as an inducer, and when it acts as an inducer, it deactivates the repressor. So when it deactivates the repressor, it changes the shape into its allosteric inactive form, so it can no longer bind to the operator. When this occurs, RNA polymerase can then go down the length of the DNA now that the pathway is open, and it can transcribe genes into mRNA. So E. coli has 3 enzymes which take up and metabolize lactose, and these enzymes are called LAC-Z, LAC-Y, and LAC-A. LAC-Z codes for beta-galactosidase, which hydrolyzes lactose into galactose and glucose. LAC-Y codes for permease, which is a protein membrane that transports lactose into the cell. And LAC-A codes for transacetylase, which its function is still not clear.